Good morning, dear students. Welcome to my channel Patel Science Academy, and from the lecture Enhancement of the Food Production. This is lecture number 8B, the second part of the lecture number 8, and the topic in this lecture is microbes in human welfare. So, dear students, as you know, this is Patel Science Academy, and this is Shaza sir. So, I request you to watch the whole video till the end so that you can learn the rest of the topic in this lecture. So dear students, in the previous lecture I have explained about so many role of the microbes in the different industries, in the formation of the organic acids, in the other household products too, as in the biogas production, in the sewage treatment too. So now from last lecture I was explaining about the role of the uh, microbes as a biofertilizer. So as you know, dear students, microbes, they also play their role as a biofertilizer too, because fertilizers are the nutrients which are necessary for the growth of the plants and thus for the productivity of the cultivated plants. So use of the fertilizer for increasing the productivity is one of the aspects of green revolution, dear students. And fertilizer are classified as inorganic, that is chemical, inorganic, biological. So as you know, this there are some living organisms, and that living organisms are being used as fertilizers in the field of agriculture, and that living organism is considered as a biofertilizer. So we are watching and considering here the some of the example of the biofertilizers from the bacteria, from the virus, from the algae, from the fungus group as a biofertilizer. So nitrogen fixation is one of the aspects of that biofertilizers. So the nitrogen fixing microorganism which converts the atmospheric nitrogen into nitrogenous compound like the nitrites and nitrates via ammonia. These nitrogen fixing microorganisms, dear students, these nitrogen fixing microorganisms also called diazotropes, and they are of two types. First is symbiotic nitrogen fixing microorganism, and another one is free living, means non symbiotic nitrogen fixing or microorganism. What is symbiotic and what is the biological or environmental or human? So bacteria, which called a nitrogen fixing bacteria, these bacteria forms the symbiotic relationship with the host plant, and the bacteria live in the nodules found in the roots of the leguminous family of the plants. For example, in the bean plants, in the pea plants, and in the clover plants, they do reside, or they are located in the nodules present in the roots of that leguminous plants, and that bacteria, because they fix the nitrogen, atmospheric nitrogen, in the root of that in the nodules of that plants, that's why it is called nitrogen fixing bacteria. And what is symbiotic association or symbiotic combination? They are students symbiotic nitrogen fixing microorganisms, for example rhizobium bacteria, for example anabina, for example frankia. All these are always associated generally with underground parts, that is the root of the higher plants. So as they are located in the underground parts, that is the root of the plants, that's why they are always in the association of that roots, that's why they are called symbiotic. For example, this bacteria, rhizobium bacteria. And what about the free living bacteria? Dear students, this free living bacteria is also known as non-symbiotic nitrogen fixing microorganism. An example is Azotobacter, Nostoc, Clostridium, Bijerinkia, and Calapsila, etc. All these are non-symbiotic because they do not live along with the root nodules. They do not fix themselves with the root of the plants. They live freely in the soil. That's why they are called free living or non-symbiotic. So you can observe this root nodules in the root of this leguminous plants family, and bacteria resides there and converts the atmospheric nitrogen into ammonia, and likewise they provide the nitrogen to that plant's roots and from plant's root it's going the upward direction. So this is that Rhizobium leguminosarum species in the garden pea plant, and you can see these are that legumes. You can see these different legumes. In these legumes, bacteria resides there in these legumes and provides the nitrogen, fix the nitrogen from the atmosphere and convert it uh, as a nutrients into this garden pea plant. So name of the bacteria is Rhizobium leguminosarum bacteria species. Thereafter the next one is Rhizobium fascioli species in the bean plants. So you can see the bean plants legumes. These are legumes, and in these legumes bacteria is stick always to these legumes of this root and provides the nitrogen to this plant. So this is about the example of the bacteria to the bean and the what we say garden pea plants. Thereafter, free living bacteria, as I told you, these highly specialized bacteria live in the soil and have the ability to combine the atmospheric nitrogen with the hydrogen to make ammonia, that is NH3. So this is that bacteria, dear students, and this bacteria combines this nitrogen with the hydrogen, and this free living bacteria live in the soil and combine atmospheric nitrogen with the hydrogen and forms this nitrogen ammonia, change this nitrogen into this ammonia and provides this nitrogen likewise to this roots of the plant. So this is about the free living bacteria, but the difference in between the free living and the symbiotic bacteria is symbiotic bacteria is always associated with the nodules of the root, and free living bacteria is always free. Thereafter, the example is of the free living bacteria is Azospirillum azotobacter as a biofertilizers. Then the next one is dear students, you can just understand this by watching this video carefully. What is happening here in this video, the nitrogen play a big role in the plant's growth as it is a part of the chlorophyll and the amino acids which make protein. So dear students, the problem is that nitrogen in the soil is limited, so when the plant uses all the available nitrogen, it will be wither. So this nitrogen is found most commonly in the air we breathe, and but this form of the nitrogen is highly inert. So dear student, what to do? What does plant do? Because it is so inert, these trees and plants cannot use this type of nitrogen found in the air. So what to do? To find out how plants get nitrogen, we need to go underground. Nitrogen from the air diffuses in the soil likewise, but the plants still cannot use this nitrogen. So along comes now bacteria, Rhizobium bacteria to be specific, and this special bacteria are able to take this form of the nitrogen and change it into the form of the plant can be used. So this form is called ammonia, and the plant can take this up and use it for its various functions. This process continues to repeat itself. To truly understand the intricacies of this process, we need to look even closer at the root of this plant. The plant releases chemicals called the chemoattractants that attracts the kind of this. The rhizobium comes running. Once attracted, the bacteria attach to the roots of the plant. The attached bacteria send NOD factor into the plant's root cell, and that makes the roots curl, as you can see here, and much like in this diagram here. The bacteria get trapped in the pocket as shown here, dear students. I hope you are getting it, and a tube tube grows between the pockets of the bacteria and main roots called the infection tube. The rhizobium travels down to the tube and start to infect the surrounding cells likewise, and this infection makes the cell in the root of the divide quickly causing a nodules to grow, and this nodules is where the bacteria live. They receive the sugar from the plant in the return of for nitrogen. This is what the nodules looks like. You can see here. So we have seen how nitrogen start in the air as N2 and how it diffuses into the soil and how it is changed into the nitrogen that the plant can use, and how bacteria creates root nodules that benefits both the plants and the bacteria. So this is called symbiotic association because both of these organisms are getting it. So dear students, I hope you have understood about the nitrogen fixation process here. So I hope dear students you have understood.
Created using Alton.
So dear students, I hope nitrogen fixation is understood by you. And in the aquatic environments like the fish tanks and the blue-green algae, cyanobacteria is an also the important free-living nitrogen fixer. You can see here the hydrolysis in the nostoc. This is the hydrolysis. Actually, dear students, cyanobacteria as a bacteria, there are many cyanobacteria are aquatic and terrestrial, free living or symbiotic, aerobic, photosynthetic, nitrogen fixing, heterocystous or the non-heterocystous form. For example, Anabina, nostoc, you are observing here nostoc. Some other example are also there, Plactonema, Oscillatoria, etc. Anabina, nostoc and the Tolipothrix are the associated with the lichens, while the Anabina and is associated with the plants like the Azula and the Cycas. So dear students, besides this there are some other examples too. The example of the fungal biofertilizers like the ectomycorrhizae and the endomycorrhizae is there. So all that we'll see one by one here. First, this is the nostoc thallus, and it is used to provide the nutrition to the plants. So these are the Anabina and Azolla plant. Anabina and Azolla live together. This is mycorrhizae that is useful as a fungus. in the field of the microbes in human welfare for the plants as a biofertilizer. So this is mycorrhizae, and dear students, this mycorrhizae is a fungus, and it forms a symbiotic association with the underground parts like the rhizomes and the root of the higher plants occurring in the thick humid forest. These are discovered by the Frank in 1885, and there are of two types, ectomycorrhizae and endomycorrhizae. So dear students, what is ectomycorrhizae and endomycorrhizae? Ectomycorrhizae, they have well developed mycelium. So as you know, the structure formed by the roots of the plants and the hyphae of the fungi, it helps with the absorption and the symbiotic relationship products of the photosynthetic in the return for the nutrients. So the detailed account of it, there are two types of mycorrhizae, ectomycorrhizae and endomycorrhizae. Ectomycorrhizae, they have well developed mycelium, and that forms the mantle on the outside of the roots, and this increases the absorptive surface area of the roots and accelerates the uptake of the water and the nutrients, that is nitrogen, phosphorus, calcium and potassium. Due to this, the plant vigor, growth and yield increases, and some hyphae of the mycorrhizal fungus penetrate into the root and forms the hartignate. So you are observing here hartignate, hartignate, in the intracellular spaces of root cortex. This is about ectomycorrhizae. Then about the endomycorrhizae, dear students, endomycorrhizae, they grow in between and within the cortical cells of the roots, and fungal hyphae penetrates the cells and forms finally branched known as arbusculus or arbuscules. So this arbuscules intracellularly and forms the vesicles mostly in the intracellular spaces of cortical cells, and hence they are called VAM, that is vesiculo arbuscular mycorrhizae. Once there was a question on this VAM in the board examination. VAM, it stands for V for vesiculo, VESICULO, A for arbuscular, ARBUSCULAR, and M for mycorrhizae. So VAM means vesiculo-arbuscular mycorrhizae. So nowadays they are described as a AM fungi, capital A capital M fungi. The plant with a VAM means the plant with the vesiculo-arbuscular mycorrhizae grow luxuriantly in less irrigated land, and the association of the VAM with the crop plants helps in the conversion of the less productive field into more productive field. So what are the benefits of the mycorrhizae? The benefits of the mycorrhizae includes it is for the selective absorption of the potassium, phosphorus, zinc, copper, calcium, nitrogen, manganese, boron and iron. It also enhances the water uptake. It induces the growth by secreting the hormones, and one more benefit is it offers protection to host plants from other microbes by secreting antibiotics. So dear students, this is about the benefits of the mycorrhizae. Do you know, dear students, nowadays mycorrhizae are classified into 8 different types, with ectomycorrhizae, endomycorrhizae, ectendomycorrhizae, orchidaceous mycorrhizae, ericoid mycorrhizae, arbutoid mycorrhizae, monotraploid mycorrhizae, and the last ophioglossite mycorrhizae. So all these are the different 8 types of the mycorrhizae. Mycorrhizae are not obvious, and plants roots without VAM, you can observe here same plants root colonized with the VAM. This is that VAM, what is called vesiculo-arbuscular mycorrhizae. It proliferates spores, sporangiospores, which are the overseeing source on proliferation and survival in the soil. This is about the role of the mycorrhizae. So this is that VAM, vesicular arbuscular mycorrhizae, endotropic. So it is not detectable to naked eyes, not short, stubby and branch root like EM, no flesh of the micro mushroom. Fungus infects the root or root hair and grow into the cortical region and penetrate cells, developing tree-like highly branched hyphae insides, as I shown you in the previous slide. That is called arbuscules, main interface for the nutrient exchange, and most species produce the large vesicles full of the lipids. So that's why it is called VAM, vesiculo-arbuscular mycorrhizae. So this is about the role of the microbes as a biofertilizers. Now we'll go for the role of the microbes in the biogas formation. Dear students, biogas is produced when anaerobic bacteria break down. So this anaerobic bacteria, actually, this play very important role in the production of the biogas. Actually, most commonly used models of the biogas plants are KVIC and IARI. The digester used for the biogas production is called biogas plant. A typical biogas plant using the cattle dung as a raw material, consisting of the digester and the gas holder. Digester is made up of concrete bricks and cement or steel, and this is a cylindrical gas holder or gas tank above it to collect the gases. Digester has a side opening, that is large pit, into which the raw material as a cow dung is fed, and the digester is partly buried in the soil. So anaerobic digestion involves in three processes, and that three processes are: first one is hydrolysis, second one is acidogenesis, and third one is most important, that is called methanogenesis. So by these three steps, biogas is produced by the anaerobic bacteria, and this biogas is actually nothing but the mixture of the methane and carbon dioxide gas. So dear students, this is that biogas, this is that biogas plant in which there is a big role of the anaerobic bacteria, and this is the influent and this is effluent. So this is about the small biogas plant in which from one side sewage, manure or plant material or cow dung is inserted, and then it is processed by the anaerobic bacteria, and then biogas is produced. So let's see all these steps one by one. So what are the basic biogas requirement? It includes, dear students, domestic waste, agriculture waste, agro-industrial waste, municipal waste, and the forestry waste, etc. So these are the requirement of this biogas production. Then the next step for the biogas production, its composition. Its composition includes 50 to 80% in the biogas there is methane. Which gas is there? 50 to 80% methane gas. Then carbon dioxide gas percentage, it is in between the 15 to 45%, while the other gases in the traces are also there. So mainly methane and carbon dioxide gas is there in the biogas. So let's see its detail one by one. Here students, the biogas production process, as I told you, it involves the three steps, and that three steps, within that by that three steps we'll learn how this plant waste material and the other material is converted into the gas. So it includes the first step as a hydrolysis first number, then acidogenesis second step, and the third step is methanogenesis. So we'll see all these three steps of the biogas one by one. First we'll go for the very first step by this bacteria known as anaerobic bacteria. The name of this anaerobic bacteria which is being used in this biogas production is Clostridium pseudomonas. This is the name of that anaerobic bacteria. You can see this is that anaerobic bacteria, Clostridium pseudomonas. So dear students, what are the role of the Clostridium pseudomonas? So before we go in the depth of that topic, I would like to explain that three steps in short. So first step includes the conversion of the polymers from that waste material. This polymers is converted into monomers. So this is the first step done by the anaerobic bacteria. So what will happen? First, polymer will be converted into monomers in the first step.